Okay, so the second major resource for bioinformatics information is housed at the EMBL or EBI, the European Bioinformatics Institute in the UK. And this resource was kind of developed at the same time as the resources at NCBI. And essentially, much of the resources that you'll find are very similar between the EBI and the NCBI. And in most cases, it's a matter of preference of of what is easier, or what do you find as the easier resource to actually access information from. You'll see once you start having a look at the two resources that there are some slight differences in the way that the data is stored, as well as the tools that are available to extract the data. So essentially, it boils down to personal preference in terms of which one of these resources you use as your main resource for extracting information. So as I said before, the EBI has very similar resources to NCBI, and it maintains the world's most comprehensive range of freely available and up-to-date molecular databases. And as well as similar to NCBI, they also offer a number of online and live training events for you to become familiar with using their resources. So let's have a look at what the main homepage looks like. So again, this homepage does undergo updates on a regular basis, and there are some changes on the new when the new web pages are updated. But this is what it currently looks like. So you'll see that this center panel is similar to the left-hand panel that we saw in NCBI. So it's where they've categorized all their resources according to the different categories, so DNA and RNA, gene expression, etc. Again, on the right-hand side on top here, we have a most popular resource section. So these are the most popular resources that I use at at EBI. And then we have a link here again to a training page. So this will contain all the information about the tutorials, online training courses, as well as upcoming live training events that the EBI does on a regular basis. So for the sake of comparison, what I've done is I've also clicked on the DNA and RNA tab. And what you'll see is the first thing that on the EBI page is they list the most popular services that are accessed for this particular category. So you'll see that one of the most popular services is something called Embl-Ebi. And this is an example of a genome-centered database or resource, and we will be having a look at that in more detail in the genomics module. And we have Ensemble Genomes, which is a more extensive list of databases dealing with different organisms, specialized databases for different organisms. And then we have a number of different databases and tools. So in this case, Classy Omega is a particular tool that's used to analyze to generate multiple sequence alignments for DNA sequences. And the ENA is essentially the EBI's version of GenBank at NCBI. Okay, so I'm just showing you the top of this page. If you scroll down, you'll get a list of the rest of the resources in terms of the databases and tools that are available at the EBI for analyzing DNA and RNA sequence data. Okay, so hopefully I've given you a bit of an overview of the two main resources for accessing bioinformatics or biological data, the NCBI and EBI. And what we're going to do now is I'm going to go through some examples of extracting information for a particular gene from both of these resources, and we'll see what information we can get back. So I want to first talk a bit about accessing data. So in most instances, for all of you doing this course, if you're going to be doing some sort of bioinformatics work, you're going to need to access information either for a particular gene, a particular molecule, a particular disease, etc. So why do we need to access sequence data? So in particular here, we're going to be talking about sequence data. So we might want to know what the sequence of a gene is. So we might have a gene of interest, and we want to get that sequence in order to do some analysis. We might want to identify variants. So you might want to know what variants have been identified in this gene and where are these variants located. What are the consequences of these variants in these genes? Have these variants been identified in a particular population? And if so, what is the frequency in those populations? Then we might also want to compare sequences. So we might have a sequence of interest, or we might want to extract a sequence of interest and then compare it to all other sequences that have been found before or identified or generated before. We might want to identify sequences that are similar to our sequence of interest, or we might want to see for a particular gene what other sequences in other organisms are similar to our sequence or our gene from our organism of interest. And then we might also want to find diseases, what diseases are associated with a particular gene or variation in your gene of interest. So these are some points when you are accessing data or searching for data. It's important to be clear what data you're searching for. So on the onset of doing any sort of analysis, first of all, define what data you're looking for in order to be able to find that data or to know where to find that data. Most of the tools have been developed to link all annotations for a particular query. So as I mentioned before, we have standalone databases, and then we also have resources that allow us to do one query search and get information back from a variety of different resources. So an example of that is, as I showed you before, the NCBI and EBI. There's a number of different databases that are all housed within the umbrella of the NCBI or EBI. And we'll have a look at some of the tools that will allow us to actually search across all these databases if we wanted to using a single query. And like I said, so as I said now, both NCBI and EBI provide what are called portals to allow you to search across all their databases using a single query. Okay, so the first example we're going to go through is accessing data from the NCBI. And the example I'm going to use is we're going to say that we are interested in finding out information on a particular gene. In this case, it's the FOXP2 gene. And in this case, from a particular organism, and it's human. So if you go to the NCBI homepage, you'll see that at the top of that homepage, there is a search box or search bar. And this allows you to search across all the databases that are housed at the NCBI. So on the left-hand side of the search bar, you'll see that there's a drop-down menu, and by default, it is always on all databases. If you open that drop-down menu, it will then it will give you a list of the individual databases that are housed at NCBI, and it will allow you then to limit your search to a particular database. So for this particular example, what we're going to do is we're going to leave it on all databases, and we're going to run the search using FOXP2. Okay. So this is the results page that you'll get back. And the first thing that you'll notice is that we've searched for FOXP2. And with the search query FOXP2, we've found results in 31 of the databases that are housed at NCBI. Okay. And what they do is they return a list of hits based on the different categories that, remember, we saw on the left-hand panel on the home page. Okay. So I'm just going to go through some of the main categories here. So the first one is literature. So this is actually using FOXP2 and searching against all the literature databases or the literature resources at NCBI for any entries with the term FOXP2 in it. And I'm not going to go into this in any detail now, but we're going to be having a look at this in the next session on literature searching. We then have the health section. So this would contain information about health and disease and clinically relevant variations that have been identified in the FOXP2 gene. And there's a number of different resources under that, and we'll speak a bit about that in more detail when we look at the results. We then have entries or matching entries in the genome section of the NCBI. So again, this is related to the genomic sequence or the genomic sequence for FOXP2. We then have hits for genes. So this would be information on what is known about the FOXP2 gene in humans. We then have information from the protein resources and databases. And then we have some information on chemicals. So what small compounds or anything known about the chemical activity that FOXP2 is involved in. You'd find that information under the chemicals tab. So I just want to go again through some of the popular databases at NCBI and some of the resources that you will get hits back from. So the gene database is basically your one-stop resource for all annotation information for a particular gene. PubMed is a database that stores extensive biomedical literature. And again, we'll be talking about searching PubMed in the next session and the next session. We then have the nucleotide database. So this is the database, a primary database that will store your sequence data for all DNA sequences, actually not just DNA sequences, DNA and RNA sequences. We also have your SNP database, which is a database of all single nucleotide polymorphisms. So these are single base changes that have been identified in different genes, in particular genes. And then we have the protein database, which is a database of protein sequences as well as associated annotations for those proteins. We then have another important database, which is a database called RefSeq. So this is a database at NCBI. It's called RefSeq at NCBI. And essentially what this database is, it's a comprehensive, integrated, well-annotated set of reference sequences. So essentially for a particular gene, we might have in the main GenBank database, we have multiple sequences for one particular gene. Because remember, the GenBank main database is where anyone is able to publicly submit data for a particular gene sequence, or a particular gene that they're working on. So what they've done in RefSeq is they've then correlated all the information for a particular gene. So they take all the information, all the sequence read information for a particular gene. They then try to identify which is the longest consensus sequence for that particular gene. And then for a particular gene in RefSeq, you will then have one genomic sequence for that particular gene. So if you are searching for the reference sequence, the most well-annotated comprehensive reference sequence for a particular gene, you would want to then get that information from the RefSeq database. If you search in GenBank, you might get multiple sequences coming back for one particular gene query that you submit. But if you want the most comprehensive, well-established reference sequence for a particular gene, you should access that from RefSeq. And in RefSeq, they have both the genomic, they have genomic sequences, genomic reference sequences, they have transcript level reference sequences, and they also have protein sequences. Okay. Another important database is OMIM. So some of you might have come across this before. OMIM stands for Online Mendelian Inheritance in Man. And essentially, this is a database of human genes as well as genetic phenotypes. So if you have any genetic phenotypes, say you're starting to work on a particular disease, genetic disease, and you want to essentially find a page that has all information about that particular genetic disease, OMIM is probably the best place to go and get that information from. So if you go to OMIM and you type in a particular disease name, you will then get back a page which is dedicated to information on that particular disease. What you can also do is search OMIM with a particular gene name, and then what it will do is it'll give you back that particular an entry page for that particular gene. And if that gene is related to a particular disease, or variants in that gene is related to a particular disease, it will give you back a list of all the variants as well as all the associated literature associated with variants that have been identified in that gene and what disease that those variants have been linked to. So if you are searching for any information about a particular Mendelian disease, OMIM is probably your best place to go and look for information, or to find information for a particular disease, as well as any variants that have been identified in that disease and the phenotypic impact. And then the final one is ClinVar, and this is a database of genomic variation as well as the relationship to any human health or human disease. So this is not only related to Mendelian inheritance or Mendelian inherited diseases, but more general in terms of genomic variation that has been identified in genes and the relationship to human health. So in the next lecture, we're going to continue having a look at the example of using FOXP2 to access information at NCBI.